Hello everyone, and welcome to this audio podcast. Interlude. This audio podcast interlude. This Quagmire interlude. This audio podcast as part of a Labonix interlude. This audio podcast as part of a Bazon scroll. This audio podcast as part of a Storm Labostics production. In this audio podcast, an overview regarding chemokine receptors and relevance in stress and coping research. So sudden stress is recognized to modify T cell trafficking and augment immune reactivity in native tissue. One investigation examined sudden pressure and the migration of T cells, connecting immunological activity to cardiovascular illness. The research explored the effects of sudden onset stress and redistribution of T cells and monocytes that prompt chemokine receptors CCR5, CCR6, CXCR1, CXCR2, CXCR3, and CXCR4 by collecting peripheral blood mononuclear cells or PBMCs and using chemotaxis assays and flow cytometry analysis. Flow cytometry investigation of T cell subpopulations on chemokine receptors expression reveals noteworthy upsurges in the quantities of CD3 positive, CD3 positive, CXCR2 positive, CD3 positive, and CXCR3 positive, and CD3 positive and CCR5 positive lymphocytes. This has been consistent with works published in around the early millennium. Sudden stress may endorse the movement of provocative cells, potentially contributing toward complications that follow stressful events. Intriguingly, these results designate that sudden onset stress brings on armament white cells, which are primed to react in trauma or exasperated tissues. Acute stresses would appear to prompt the recruitment of circulating immune cells. Consequently, it would be reasonable to examine chemokine receptor trafficking to distinguish CD4 positive and CD8 positive T cell subsets in reaction to stressful life events and coping responses in individuals with chronic illnesses. Research has not evaluated the connotation between stress and cellular adhesion molecule expression following the adoption of a particular coping strategy in chronic illness. Chemokines deliver particular signaling to T cells for extravasation from the blood. In count CMS cellular adhesion molecules, cellular adhesion molecule amine of L-selectin indicates partisan cell conscription by improving binding to nearby ligands. In mammals, evidence has suggested a dynamic relationship or association between psychological stress and cellular expression of adhesion molecules on T cells with escalation of leukocyte functional antigen LFA1. Accepting the crucial role of conscription of pressure in arbitrating leukocyte trafficking, one investigation examined the effects of strain on chemotaxis and cellular adhesion molecule expression. Reactions to five chemokines and stromal cell derived factor SDF1 regulated on activation normal T expressed and secreted chemokines, for example, in around rantes, IL-8, interleukin 8, IL-8, and monocyte chemokine chemoattractant peptide MCP1 were determined with the proportion of lymphocytes expressing L-selectin and macrophage associated antigen or MAC1, and assay of circulating concentrations of L-selectin Rantes, which connects to CCR1 and CCR5 receptors, and MCP1 binds to CCR2 receptors, is to preferably to attract T versus B cells. However, CCR5 receptors also exist on B cells. Interleukin 8 binds to CXCL8 receptor primarily attracts B cells and not T cells or monocytes. SDF1 binds to CXCR4 receptors, also powerfully attracts B cells, but also attracts monocytes, and CXCR4 receptors exist on T cells. That draws this audio podcast to a close. This audio podcast is part of a Quagmire interlude. This audio podcast is part of a Labonix interlude. This audio podcast is part of a Bason scroll. This audio podcast is part of a Storm Labonix production.